Thank you, Dixon, for the very kind introduction. Hello, everyone. My name is Andrea. I have been working with IVL, with Bibles for a little over a year now. And today I will share the methodology, mainly behind surveying oyster populations in Sweden using towed video. This project has been carried out in cooperation with Mats Lindegaard and his team at the University of Gothenburg, and Anne Laugen at the University of Oyster. So we all know that oyster beds play a very important ecological and economical role in marine ecosystems all over the world, but they are also one of the most endangered types of benthic habitats. In Europe only, there's been virtual extinctions of the native European flat oyster, Ostrea edulis, and that has been documented in many different areas. The major reasons for this are overexploitation, pathogens, habitat destruction, pollution, and the introduction of new species. So because of the important role that oysters play in marine ecosystems, they have been identified as priority habitats since 2003 by the Oslo Paris Commission. In order to manage and protect these habitats, you need quantitative data on the distribution and the quality of species in benthic habitats. And even though it is known that they can have dense populations in some Swedish parts, the prevalence, abundance, and environmental preferences are not that well studied or that well documented. And for that, to gather this data, video-based methods have been frequently used because they are particularly well suited for surveying sensitive benthic environments, such as the flat oyster bed. So tow video, it's basically a sled that we drag along the bottom on a predetermined transect. It's composed of a camera that is pointing directly towards the substrate, and then two buoys just to keep it up. So what are the advantages? Is that it's cost efficient, and it's a very non-destructive sampling method, which means that it will generate very sustainable management and conservation practices. Because you're recording video, it generates permanent data-rich records that can not only be used for studying oysters, but also other organisms like different fish. We have nudibranchs, crabs, sea stars, and you name it. But this can also be used to re-examine your own results. So what we have done is that we have surveyed 436 sites, which are divided into 5 different areas. The ones that we have called area 1, 2, 3, and 4, and the Coster National Park. These range in depth intervals between 50 centimeters down to 10 meters depth. And now I'm going to show you a little bit of a video of how fieldwork works. So basically, I'm holding the sled, which is mounted with a camera, and then you just drop it at the bottom. And then we are right there in our GP, or with our coordinates. We drag it along the bottom in a 40-meter transect at about 0.4 knots to make it more or easier to make the counts and stuff that I'm going to talk about in a little bit. So when you get the video in your hands, this is what you can see. So we divide the 40-meter video transect into 2 smaller transects, and that way we get 10 thumbnails per transect in which we can obtain vegetation cover data and the substrate data. And then throughout the entire video, we will count the live flat oysters, Pacific oysters, and blue mussels. So just for if anyone is not familiar with the differences between the flat oyster and the Pacific oyster, the flat oyster is more round. In general, it's flat, and it has a bit more serrated edges. It's not too easy to see here, but you will see it in the next slide. And the Pacific oyster is very wavy. In here, it's, I think it's very clear to identify the difference between the two. But as you will see, what we get in the field, this is one of the easiest images I could find from a video, actually, when we try to identify. Flat oysters are generally quite round, and you can see the serrations that are not as big as on the Pacific oysters, like here. It is important also to, when we do this, to be able to identify which individuals are dead and which ones are alive. That is perhaps one of the challenges for a human, but I think if we talk about automating this process, it's also very challenging for a computer to do that. This is a little bit more difficult, a more difficult environment that we get, because there's a lot of shells and there's a little bit of algae on top of the shells, whether they're alive or not. But here we can see both Pacific oysters and flat oysters. This is perhaps even more difficult to identify all the alive individuals. But we have, with the team, we have trained a lot. We have also agreed upon what we will count as alive and what we will not. So as I said, some of the challenges are distinguishing between live and dead oysters. Ideally, it would be great to be able to automate this process, but it will take a lot of machine learning. The computer will need, as you can see, the pictures, the images will look very different depending on the type of environment that they're in, on the substrate, on the vegetation cover, and the turbidity also of the video. But it's not bad to dream. So there's been a paper before that compared the video transects compared to actually doing it in the field by hand. And there was a strong correlation between field and video analysis, even though the video analysis underestimated the total abundance of the living oysters by 20%. And that is perhaps because when you grab something, an oyster from the bottom, as you see it, you can be like, oh, it's probably just one oyster. But if you grab it, then there's a bunch of oysters altogether. This research also showed that it's repeatable within and among observers with no bias or no clear bias when identifying living or dead oysters. And the conclusion was that this code video method was recommended for future monitoring of oysters and perhaps other threatened habitats as well. If you want more details about this, you could probably check this paper. And recently, a new published paper by our team discovered that both the native flat oysters and the Pacific oysters are widespread at depths shallower than 5 meters, but they have slightly different depth preferences. Nonetheless, they have a significant overlap in location, depth, and habitat preferences. And that at least for the flat oyster, which is Ostrea edulis, the substrate that was the most important predictor for the presence of these oysters was gravel. And if you have over 15% of gravel, then you would most likely have a high density area. And these high density areas are super important for the entire population in general. And that is why the conservation is very valuable to be on the side of the high density bed. This is the paper that you can look at for further details. And yeah, if you have any further questions, you can contact Åsa, I think she's here, or myself, or of course, any of the researchers in the previous papers.